Welcome back. I hope you had a productive break. Just a reminder of some things that are going to be happening later on today. There will be one more lab exploration at the end of the final session today. We encourage you, anything that you haven't seen, any buttons you haven't collected, we're definitely interested in you filling out your dance card, shall we put it that way. One thing that relates to housekeeping, some luggage that was in the lobby, there was a coat check area sort of down in the lobby, impromptu. That's all been moved up to the 6th floor. If you had luggage that you kept down on the 1st floor, it's now up here on the 6th floor in the coat check area. So if you have any concerns about that, you can go and check in the coat check area to make sure that your bags are there. But there is nothing down on the 1st floor, and everything that was on the 1st floor is now on the 6th floor. So I think we've accounted for just about everything. Let me begin by telling you a little story. This is going to be a very special session here. And as you know, I am a member of the media, and this is the Media Lab. And the media that I'm a part of goes back a long, long way to something we describe, maybe you've heard of it, as radio. Radio, of course, the encryption of RF signals that are then decoded in an analog fashion, analog that precedes digital, an analog fashion, and then rebroadcast through little sets that are also called radios. So the interesting thing about radio is that radio, what are you doing? Radio refers to what it is I do for the last 30 years. All right, this is ridiculous. Who's running this? Okay, oh, come on. Oh, this is not fair. What's happening here? What you're seeing is something that we are going to roll out in this particular session today. If you'll notice back in the audience, our fearless leader, Joey Ito, is what we're describing as the Google jockey of the sessions of this session and the afternoon. What we are going to try to simulate here in this room, and he's got some compatriots. Will you all raise your hands, please? Those were the individuals who were harassing me. There we go. The idea here is to simulate a single mind's response to a conference like this by using the resources of the web in real time. You hear content, it reminds you of things that you've already heard of, it reminds you of references that maybe you have experienced in books or lectures in the past or art experiments or a science class that you took a long time ago. Your mind is doing the search and the associative sort of comparison to everything that you're hearing. We're going to try to simulate that live and in real time as Joey and his team basically curate a Google stream that will come from references that you hear in the presentations, but particularly in the panels. So at a certain point in the 2 panels that we have later on after this session, and also at the end of the day, you'll be able to see a stream of the content that will come from information that they search, references that they see, but you can participate as well, and you can participate online as well. Give me the hashtag, guys. What's the hashtag? Media Lab IO. That's the hashtag to participate. You can email directly. Is there a way to email directly? Nope. Okay, so it's just using that hashtag, direct, oh, that reminds me of, or you should check this out. And I'll be doing a kind of a mind meld with the team up there. If you hear me say, what you got, Joey? You got something for me? It'll go up on the screen, and he'll raise his hand, or one of the members of his team will raise their hands when they have something that's of particular interest, and we'll put that on the screen, as well as simultaneously sort of experiencing the stream as we talk. So that's a feature of the presentation today, and we wanted to roll that out in this particular session. So we're going to begin now exploring the mind. We've talked about complex processes inspired by nature, nature confounding us with its level of complexity. Nowhere is that complexity greater than in the human mind itself. And so a series of presentations now on how technologies of today inform, confound, or deliver to us insights about how our complicated identities that reside within this body and brain. And to begin, a faculty member here at the MIT Media Lab, I'm sure you're familiar with her work on affective computing. Please welcome Rosalind Picard.
Thanks. It's a pleasure to be here. I've spent most of my career working on technology as an electrical engineer and a computer scientist. Technology that is really characterized by being rational, objective, measurable. And when I first learned about emotion, I thought it was the exact opposite of that. Used to think that. Now we know that that is flawed thinking, and I'm pleased to be here now giving you a few highlights of some of the technology in progress that is bringing emotion into the space of things that are rational, objective, and measurable while deepening our understanding of what it means to be human. Let's start with a question for you. What do you think is the most common emotion that technology elicits today? Frustration. All right, good answer. I don't know if this hint helped. Okay, let's see an example of us eliciting that in an experiment. Need the audio up, please.
I am sorry. You seem to be having difficulties getting through this form. Would you mind looking at the camera and share your thoughts on how to improve the form?
This one sucks.
Okay, so that was one of many experiments we do to elicit frustration. We also, of course, are interested in creating delightful experiences. And the experts will tell you that you can recognize delight by looking for two things on a person's face. Not just the smile, the AU12 or lip corner pull, but also the AU6, the cheek raise, the little wrinkles of crow's feet around the eyes. And it used to be that we would say that if you saw both of those, that meant the person was truly happy. Whereas if they just went, cheese in front of the camera and only smiled with the mouth, that wasn't a true happy smile. I used to believe that until we started working not so much on acted and posed data, but working on spontaneous real-world affective experiences. Let me show you where this particular face came from. Kind of a coping mechanism. Now notice the speed of the smiles here and that they involve the eye wrinkles too. These are smiles that are not delighted smiles. He reported feeling enormous frustration. When we, in fact, captured facial expressions, smiles from people experiencing truly reported delight and true frustration, these peak smiles look so similar. When we gave them to a group of people to tell them apart, they were random. About 50% accurate at figuring out which were delight and which were frustration. I failed the test myself. Turns out that it's very hard for both people and machines to learn to recognize true affective expressions from faces and from the information that we exhibit around us. But there's some cues some of us learn that make some of us better than others. This is just some data where the green bars represent the recognition accuracy of several different machine learning algorithms on acted data of frustration, delight, and neutral. And the red bars represent the machine accuracy, which is close to random, on the same data, but when it came from spontaneous situations. So it's a very hard problem to recognize emotion in the wild, in the real world. Now I'm going to skip over a ton of technical detail and just hop to some results here. This paper has just been accepted to IEEE Transactions Affective Computing. You can get it online, work based on Hook, Dan McDuff, and myself. We are making headway. It's hard work, but we have now got computer machine learning algorithms that are able to do as well as the human on the frustration smiles, well, even better than the human on the frustration smiles, while still doing as well as the human on the delight smiles. So we're making progress, but this is just two kinds of smiles. We still have a whole lot more we need to do, and bringing people in to the lab just doesn't scale. Academic researchers really haven't made rapid progress in this area, in part because they've depended upon bringing hundreds of people into the lab. What we really need to learn the full space of some 10,000 combinations of movements on the face is probably hundreds of millions of samples, maybe even more. To do that, we're going online, and we have launched last year the first collaboration that put affective of collection and aggregation out into the cloud. So I'll show you a sample of that. This was a joint project with Dan McDuff and Rana El Kalyoubi here with Affectiva, launched on the Forbes website. You can go there and try this live. Here's a person who opted in to have her face shown publicly, while the left shows the aggregated data from lots of viewers who watched this ad. I don't know how many of you saw that when we launched online. Now we also have a lot of other facial expressions online, not just smiles. It's interesting, though, to see things like the yellow, the orange and green lines at the top are people who had said they had seen the ad before, and notice how much more they're smiling than the first timers. This ad was so effective that people were actually even happier the second, third time, and we don't know how long after, to see it. And they're even smiling in anticipation of the punchline at the end. They're enjoying it so much. And this was considered actually the best ad of last year.
So my research focuses on building.
This is my collaborator, Rana El Kalyoubi.
Emotional states through their facial expressions. We've built technology that looks at people's facial expressions. It identifies the main feature points on your face, like your eyes, your eyebrows, your nose, and then it tracks how they move over time. When I smile, the green smile track goes up, and when I nod my head, the head nod track goes up. And when I shake my head as in disagreement, you see the pink line go up.
We use thousands of videos of people showing agreement, showing disagreement, in order for the computer to learn what it looks like, liking, concentrating, disagreeing.
Taking this technology online opens a whole lot of new applications. Almost everybody with a computer today has a webcam, or a lot of smartphones have cameras too. So if we enable people to opt in, turn the camera on, they can share their experiences online. It's a more sophisticated version of the like button on Facebook.
Imagine a situation like recently in Egypt where Mubarak was talking as if he believed the people still liked what he was saying. Imagine if the people could easily and anonymously have opted in, flipped on their camera while watching a speech, and expressed their satisfaction and dissatisfaction with a simple facial expression. That information aggregated going up is very powerful data. Now, my close collaborator, Rana El Kalyoubi, is from Egypt. She was in Cairo during the big uprising, and she was not in Tahrir Square. She stayed home where she felt safer. But she said that she thought that maybe Mubarak didn't think 1 million people was a big enough response to actually believe that people were upset. There are 80 million people in Egypt, and most of them were not there. If we could enable all of them to have the opportunity to safely and anonymously opt in, and while they're observing a speech, to give their nonverbal feedback and join that conversation, that would be very powerful. Now, I've shown you some examples of face reading and aggregation of that online that in the future is going to enable new forms of communication among very large groups, as well as individual feedback for experiences that you might have on the computer. But what about the stuff inside that we may or may not show on the outside? In particular, we work a lot with people on the autism spectrum, many of whom may have very strong feelings inside. And in fact, when those feelings become almost overwhelming in an autonomic, excited, aroused state, their motor system may shut down and they may show nothing on the outside except looking like they need stimulation, when in fact it might be the opposite. Autism now affects 1 in 88 children in the US, 1 in 54 boys. The emotional signal that we want to get that's not captured by the face is what's called the arousal dimension of emotion. So this goes up with things that activate you. When you are excited in an audience, we've seen it, for example, go up when there's laughter or live interaction, and unfortunately go down pretty reliably when there's PowerPoint. We've been looking at it in classrooms, too. It especially is correlated with memory and attention. So it's a very important signal to look at for a lot of reasons. We've been particularly interested in it when people have trouble with behavioral emotion dysregulation. Those of you who have teenagers and toddlers know what I mean. And what we needed to do, though, to measure it in the wild is get it off of the way that it's traditionally measured, which is on the fingertips with electrodes and wires. This, we have done a number of innovations you've seen here over the years, where now it's available, for example, in wristbands that I'm wearing here, which are now commercially sold by Affectiva. Full disclosure, Rana and I are co-founders, and I'm also chief scientist and chairman of the board. This technology now is enabling us to measure sympathetic nervous system activation on the fly. Now, this is something inside all of us that is regulating all of our organs. It's sending signals to your heart to cause it to beat faster. Your parasympathetic nervous system sends signals to your heart to cause it to slow down. People often ask us, why are you measuring skin conductance? Why don't you just measure heart rate? Heart rate, when it goes up, we don't know if it went up because of the sympathetic kick or because of somebody stepping on the parasympathetic brake and pulling off of the pre-revved heart. The skin is purely innervated by the sympathetic nervous system, so we get a pure sympathetic response measuring it. Here's an example of a girl on the autism spectrum who has limited vocabulary. She really can't tell us what's going on inside her, and yet we can see her data streamed in real time. The right edge of that blue window at the top is showing you her arousal level live while she is engaged in the activities on the left. We saw it peak as she got on this swing. Cognitive, emotional, physical exertion can cause the sympathetic arousal to climb. And while she swings and calms down that nice repetitive movement, we see it declining nice and peacefully. With this technology in the future, we'll be able to understand things about our loved ones that we weren't able to see before, things that calm them, things that stress them. And I've already seen nice interactions change and behaviors change when people share this information with others. And suddenly they believe you when you say that something affects you in a certain way. When you go out in the wild measuring emotion, you often find surprises. And one of the biggest surprises and sort of deeply exciting ones that has happened here was when one of our UROPS, undergrad researchers, asked if he could borrow the sensors, early prototypes of them, for his little brother on the autism spectrum to wear over Christmas break. And I was looking at the data later, and I saw this one gigantic peak on one side, one wrist, and not on the other. And I thought, oh, darn, sensor's broken, right? Peaks just don't happen that big. I zoomed in to try to figure out what kind of malfunction it was, and all the data actually looked good before the peak and after the peak. It was just this enormous peak that I've never seen anything that big. Made sense. So I call up the UROP at home and I say, hey, any idea what happened on December 26th at 4 o'clock? And he says, I'll check the diary. He goes and checks his little brother's diary, and he says, that was 20 minutes before he had a seizure. And I went, wow. And I called another UROPS dad, Joe Madsen over at Children's. Hi, Joe Madsen. My name's Rosalind Picard. Is it possible that a person could have a sympathetic surge before a seizure? And Joe got very interested in this and said, yes, probably not before it actually starts in the brain, although maybe, but possibly it could happen before people on the outside see it, see what's going on inside. So we began a collaboration. I began to learn a whole lot of interesting things about epilepsy. One very surprising fact is that seizure-related consequences kill more people every year than breast cancer. 1 in 100 people have epilepsy. It's very common. Surprisingly, not anywhere near as well known, not as good a PR campaign, I guess, yet. Now, we undertook this collaboration where we collected EEG, electrocardiogram, and electrodermal activity, EDA, off of both wrists from nearly 90 kids around the clock, 24/7, while they were in the hospital being monitored as candidates for brain surgery. These were kids with very serious repeated seizures. And what we found when we compared the EDA to the doctor's labeling of the data blind to ours, they were labeling it just looking inside the brain, we were labeling it just looking on the signal on the outside of the skin, is this very high correlation. In fact, it's more than that. We were able to use our machine learning and pattern analysis to build an algorithm that using only the signal on the outside of the wrist can detect information that tells you when the seizure is happening with 94% accuracy. So we now have a wrist-worn seizure detector that is basically telling us what's going on inside the brain during these times. It's not going to replace the EEG, but the high correlation allows us to use it to detect these events that previously were not detectable in this easy way. We also have a very exciting finding getting ready to come out, and it relates to one of the suspected causes of death from seizures. People don't know what, other than hitting your head and drowning, why so many people have a seizure and then seem to be alive and fine, and then they die after the seizure. One of the hypothesized reasons is that the brain waves sometimes, actually, it's like the brain from being overactivated during the seizure goes to being underactivated, like something suppressed it. And the duration of the time those brain waves are suppressed is a hypothesized biomarker for this sudden unexplained death, unexpected death from seizures. This is a public talk. I can't say this till 4 o'clock today when it will be public because of neurology medical journal embargoes, but in neurology today, we will be announcing a very exciting finding. I'll invite you to come and talk to me about it privately or hear about it publicly this afternoon, relating what we can now sense off the wrist to something we think is very significant in terms of helping with the health and hopefully saving lives for these individuals. In closing, I'd like to mention one last future piece of work that is getting ready to happen with Peter Weinstock at Children's Hospital Boston and Cynthia Brazil and myself. Peter is describing emotion now as the 4th vital sign and talking about how, as a doctor, it's critical when somebody comes in to assess their pain, their stress, and treat that as well as the other symptoms of disease. We are now able to measure pain responses with this sensor as well as we start to measure emotional information. Different kinds of signals we can pull out and interpret, and this sympathetic activation goes up with pain. So we're going to be looking next at the children's pain in the hospital, which experiences decrease that, and what we can do to improve their health and their experience while they're there. We're also collaborating with neuroscientists looking at the different circuits in the brain that send signals differentially to places on the body. We used to think that the arousal and emotion was just a general thing, hits your whole body at once, but it turns out it's measured differently and has some different information at different positions. So we're very excited about mapping those things out as well. So in the future, we will see a lot of different new tools and technologies that allow us to measure and communicate emotion in new ways. And I hope you'll join us in helping us invent new ways to make these most effective for improving people's health, people's opportunities, and their experiences, online, face-to-face, for all people everywhere. Thank you.
Hold on one second, Roz. Here, just take a step over there so we can get in the middle for the camera shot. Here, very good. Just a couple of simple things. And I always find your work fascinating. Yesterday, Todd was talking about the amount of data that is apparently in the human voice in terms of its ability to communicate things like emotion and what's going on. Cesar talked about big data, large data sets. How much data do you think is coming off of a person in the kinds of effective parameters that you're beginning to measure? How much data am I radiating, or is any given person radiating? And then how much data do you think you need in terms of these 200 million subjects to create the kinds of algorithms that give you a reliable model for what we think is happening in any given emotional state? How much data are we talking about?
Great. Oh, great question. In terms of what's coming off, some people have argued that up to 90% of what we're sending out is affective nonverbal information. Posture, gesture, voice, face, perhaps some of our hormonal, olfactory signature, and a whole lot more. So there's a lot being perceived that we actually don't know how to quantify yet. We don't know how to digitize all of that information yet. In terms of number of people to get the algorithms accurate, more is almost always better. So even once we get millions, like right now we're up to tens of thousands. We have the best smile recognizer. It's been tested in China, India, Brazil, all over. And it's in the high 90s in accuracy, because we have a ton of data on that. It will continue to incrementally improve. In fact, we're trying to discern different kinds of smiles. But the other facial expressions, until we get lots of examples, and we made the contextualized. We can't predict that accuracy trajectory, but usually more is better.
Your statistical results are impressive, yet how confident are you that what you're measuring and the algorithms that you're developing for measuring these emotional states are similar, or perhaps they're extraordinarily dissimilar, to what I'm doing in trying to read your emotional state right now? Am I looking at all the data that's coming off of you? Am I filtering things? Am I taking a predictable vocabulary of a couple of characteristics and making a conjecture? What am I doing that's either different or the same than what you're measuring with machines?
I'm tempted to have a comeback here because I know you're married and you have a wonderful wife. Most spouses miss a lot of information, right? I think when we tune in...
I've had this experience, yes.
Yeah. So,
are we going to go there? Are we really going to go there?
When we want to, we can tune in and we can read things we can't even put our finger on, right? But when we don't tune in, we can miss extraordinarily important information. One of the things we're excited about, for years we've been building these wearable glasses that we've been putting cameras and hooking to our algorithms. I'm really excited now that a couple companies, Google, Apex, others are starting to come out with glasses that we can start to gather more of this information in real time, real world social settings, and start to see how much of that is answering that question you just asked and what is still missing.
So then the technologies you're developing, would they supplement my pathetic, as a male, ability to read emotional states in someone else? Would they accumulate, or would they conflict in some way? Do they actually increase noise in the equation and actually make it harder for me to determine emotional states?
It's interesting. So what will happen when my technology tells me that you're bored by what I'm saying, but I like to think that you're interested, right? So we might have a conflict there. I think we're going to have to, follow that conflict and see what the behavior is that ensues, and then learn how to trust the two channels of information based upon what happens.
We're confident that the brain can handle additional channels.
The brain can handle additional channels, although the interface for this is a real challenge, right? Because if I'm streaming those graphs, in front of my glasses while trying to interpret your face, that's overwhelming. So we've done things like little red, yellow, green light feedback. Red, Roz, stop talking. Green, keep going. Yellow, John's looking like maybe I should slow down. Things that red.
Yep, there we go. Right.
Yeah, so things that basically help us to reduce that information to something we actually can interpret on the fly.
Finally, on your Egypt example, which was very, very compelling, if it was possible to direct a mass aggregated scale of emotional data regarding a political event that would bear directly on the judgments made by the people of a nation for its leader, what gives you the idea that any political leader would be, under any circumstances, interested in that information at any time?
Well, in the U.S. re-election, but overseas, I agree. Some of these dictators, I think it's going to be critical that maybe we protect IP addresses if people in a certain village are in dissent. We're going to have to be very careful because we know that they care. The question is, do they care in a way that's good for the people, or do they care in a way that is only self-centered? Is
there a quality of the data that overwhelms whatever inhibitors the dictator might be putting up? In other words, is there an unmistakable, it is clearly the case that you are hated and loathed in every way, and that begins to have its own sort of persuasive power, even overwhelming the existing structures of a state apparatus?
I think that's a great question. I have an answer for a different version of it. We saw people making movie trailers, and they read the audience feedback, and sure enough, the audience was showing signs, lack of interest falling off at a certain point. And the guy who took that data to his boss said it was so nice to take the data to my boss, and that was really credible, instead of just taking sort of my opinion or questionnaire data, which we don't believe. It's sort of the shoot the messenger. He didn't like taking the verbal data, but when he took the nonverbal, more objective data, he felt like that was something that didn't bear poorly on him.
So objectivity has power.
Yes.
That's a hopeful sign. Thank you very much, Roz.
Thanks, Tom.
A radical thought now from one of the most exciting personalities at the Media Lab. Ed Boyden will now tell us some of his latest work, which describes how, and go with me here, the brain, far from being an impenetrable barrier to the understanding of our human identities, in fact, if you look at it carefully and you look at it in the right way, invites us to collaborate, invites us to find ways to connect and to go deeply into how the brain is working. Please welcome Ed Boyden.
Thank you. So our group at the Media Lab works on neurotechnology, technologies that help us understand how the brain works, but also that help us figure out how to fix the brain and the computations that it performs when those computations aren't what we want them to be. And part of why we are pursuing this is because of the intricate circuitry of the brain. Over the last 100 years, neuroscientists have really had a bunch of results that help us figure out some of the properties of the brain. In particular, the fact that it's an incredibly densely wired computational circuit that's made out of hundreds of billions of electrically computing cells called neurons. One of our hopes is that we can start to understand this circuit at a level that lets us actually engineer its function to fix problems when we detect them. But to do that, we need fundamentally new tools, and that means opening up new technologies and figuring out how to build them. Now, we all wish that we could fix the brain in the same way that you could probably reprogram a piece of computer code when it goes into a non-working state or there's a bug. But part of the problem with the brain is that the elements that make up the brain are just so incredibly diverse. In fact, they come in thousands, if not more, different kinds. They differ in their shape, the molecules that they are made out of, which enable them to compute, and how they're connected. And so part of the problem is we don't even know the exact number of kinds of cell in the brain. It would be great if we could get a parts list for the brain. And of course, it would also be nice to know if we can figure out how all those parts work together in real time. And the last point is an important one, because the temporal properties of the brain are what make it particularly difficult to deal with. We need technologies that allow us to observe what's going on in the brain and to fix what's going on, to control the circuits in the brain at the time scale at which the brain operates. And so these cells, which might receive information from tens of thousands of upstream partners and send information to tens of thousands of downstream computing elements, are operating at the millisecond timescale. The pulses that they fire, the electricity that they work with, is such that, in theory, you can imagine them computing and exchanging information each hundreds of times a second or more. And remember, in your brain, the very organ that's letting you listen to these words and turn them into information and symbolic representations and mediating emotional or non-emotional responses, as it were, to the content, is the kind of thing that occupies these hundreds of billions of cells all the time. Now, part of the goal then is to really figure out, can we obtain this sort of parts list for the brain? Can we actually develop technologies that allow us to go in and figure out all these computing elements so that, much like that computer chip that I showed you earlier, we can actually go into the brain and look at these pieces and say, oh, we know what that does, and we can try to figure out how it's going to play a role in the circuit. But of course, having a list of parts isn't enough to know how these things function. Ideally, we could also figure out how they play roles in the circuit. And so I'll tell you also about how we're developing technologies that allow us to turn on or off different cells within a circuit, ideally at arbitrary scale, so that we can figure out, for example, how a cell plays a role in a computational process. If we delete a cell just for a millisecond, let's say, can we figure out what it's necessary for? And if we drive information into the brain, ideally at the resolution of intact neural circuits, which, to put into perspective, a cubic millimeter of your brain has perhaps 100,000 cells with 1 billion connections between them, we need to figure out how we can address the information processing and actually control the processing in there to fix the computations when they go into a bad state. Now, to develop tools, we have to kind of go beyond, I think, what an individual person working in a dark basement, how I spent my graduate career, can do. And that means looking beyond neuroscience itself. And we have done this. We've actually looked to other examples of human endeavor. We look at other examples of parts of the world, such as the natural biodiversity of the world, to look for molecules that can do things that we cannot figure out how to do. And finally, we're also looking at how we can engineer collaborative infrastructures that allow us to work with people, engineers who work on everything from nanotechnology to computer science, and also different problem domains from Parkinson's disease to Alzheimer's, in order to really figure out how to build teams and to work with others in strategies that allow us to confront some of these intractable brain disorders. And intractable is not an appropriate word. Over 1 billion people suffer from some kind of brain disorder that makes them seek medical attention. If you look at the list here, there are some of the disorders that are untreatable. And not only are they untreatable, when there are treatments, they tend to be partial and have side effects. To make things worse, a lot of these disorders will not only change aspects of our life, such as how long we have to live, but they can also alter our identity and change who we are. Now, we talked a lot in the beginning of this talk about the need to control circuits. And of course, there are methods for controlling circuits to fix them. A lot of the disorders on the previous slide, the symptoms of which at least can be treated in some regard with pharmacology. But if you think about the circuit that we showed in the beginning with all the cells of different kinds, with different molecules computing at the millisecond timescale, one could well imagine that if you're putting in a drug that will modulate many cells, you might miss the ones that you really want to hit, and you might actually end up modulating the cells that you don't want to change, and that can result in side effects. So really, we want to get back to these ideas. How do we get the parts list of the brain so that we can figure out the molecules that make them what they are and to figure out how they contribute to the circuits there? And then how do we fix them? How do we turn on and off the cells that contribute to a given computation in the brain? So I'm going to tell you a little bit about one project here that's new. And one of the ideas that we've been trying to pursue is, well, what would we really want to do? Could we build a robot that could really go into the brain and do the ideal experiment, which will allow us to go into single cells, harvest the molecular content so we can figure out for example, what drug targets we should go for next, or tell us what the shape of the cell is so we know where it projects, and also ideally allow us to record that cell's electrical activity all in an automated fashion, because of course we can do this as neuroscientists, but it's a painstaking art form. And so to do this, we started a multi-lab collaboration, including our group and also Craig Forrest's group at Georgia Tech, to try to figure out, could we build basically a robot that could do nanosurgery on a living brain or in living tissue? For example, you can imagine that the idea of figuring out what the cell types of a piece of tissue would be of great importance if you wanted to deploy personalized medicine remedies in cancer, for example. And so the idea here, I'll walk you through the idea here in this slide, and then we'll actually look at the robot, is to take a microneedle, something with a tip that's about a millionth of a meter in diameter, and to lower it into the brain, and then ideally to stop it on a cell, a fragile 10-micron diameter cell, whose membrane is nanometers wide, and then ideally to stop on its cell and interrogate it, figure out what it's doing, figure out what role it plays in that circuit, and of course harvest the molecular contents so that we could figure out what molecules these cells bear, and that then is of great importance for understanding the brain, but also, as I mentioned, finding better drug targets so that we can develop better side effect-free drugs. And so we actually have built this. So this is a device here that contains a microneedle that can go into a living brain or a piece of brain tissue that might be the kind that's removed when a neurosurgeon takes out a piece of the brain for somebody who has intractable epilepsy or cancer. You can see the example here, a little needle that indeed is able to go into the brain and do those things that we talked about. Now, there are people in the world who can do this in the living brain. Maybe a handful of groups have this capability, but you have to practice a lot, right? You have to know when to stop this little tiny device on a cell, with its 7-nanometer thin membrane, and it takes a lot of training to do that. And so now we'd like to industrialize this process of analyzing the parts list of the brain and to figure out how brain states change at the single cell level in a very high throughput fashion. And so if you want to see how we're scaling this up to larger and larger scales, you can come by the little area we have on the 4th floor and see how we're doing that. But suffice it to say that we're already able to use these machines to go in and in an automated fashion harvest the molecular contents of cells, which we can then analyze biochemically, and also to inject, for example, chemicals like dyes that'll let us see what the shape is. So we're very excited about this idea about the parts list of the brain, which brought us into new collaborative endeavors, as well as into new attempts to figure out how we could mimic the ideal human by building a robot that could go into the brain and do all these nanosurgeries for us. But some things are too hard even for us to figure out. And so for that, we have to turn back to the tree of life, to the natural world. And this has been an area that we've been working on a lot because, as I mentioned earlier, neurons are electrical computers. And so if we can actually control them, what we need is just something that can convert energy that we can control into electricity. And so throughout the tree of life, there are all sorts of molecules that are photosynthetic. They convert light into electricity. We put them into neurons, and we can turn those neurons on and off. Basically, the idea being, if we could convert light into electricity, electricity is what makes the neuron compute. Of course, you also have to put light into the brain. The brain doesn't feel pain. And so in animal models that are commonly in use throughout the entire field of neuroscience, we can also put optical fibers into the brain that allow us to get light into deep targets. So let me give you an example of one such path that we've taken. It turns out that plants, funguses, bacteria, all sorts of organisms have these photosynthetic molecules. In this case, you see here a single-celled green algae, which has in the back of the cell, which allows it to steer towards light, a little eye spot. And this eye spot converts light into electricity because that's how it knows how to steer towards the surface of the pond in order to photosynthesize better. Now, we were very lucky. This little eye spot contains basically little solar panels in them that convert light into electricity, just the magic trick we needed in order to put them into neurons so that we could turn those neurons on and off with light. So what we need to do, and what we did, was to take this piece of genome that encodes for this light-sensitive protein, this solar panel, if you will, and take it out of the cell and put it into one kind of neuron in the brain. Now, we don't have the full parts list of the brain. That's going to take some time, and also a lot of collaborators. But what we can do is we have a handful of well-known, characterized cell types that we can actually go for. And by using tricks such as in the field of gene therapy, we can take this piece of DNA, put it into a special vector that will target one cell but not others based upon the molecular descriptors thereof, and make that neuron now like controllable. And so we and many groups and partnering groups have developed a variety of strategies for getting very specific targeting of single cells so that these genes can be taken up, and then they'll express these little light-sensitive proteins all over themselves so that when you shine light on this neural circuit, the cell that expresses the molecules will be activated, but the neighbors that aren't targeted won't. So let's play out what we would like to do in an example scenario, and then we'll show you some data. So let's imagine we have a piece of brain here, and although we don't have a parts list, we have a handful of neurons that we know change in neural disorders. So for example, in schizophrenia, a disease that is amongst one of the biggest incapacitating disorders one can have, there are many cells that atrophy, little interneurons, inhibitory neurons that silence other cells in the brain, where if you could jazz up their performance, maybe one could try to remedy the shape of what the neural circuit is performing. And so we can target those cells using a virus, and then when we shine light on the neural circuit, those cells will be actuated and not their neighbors. So let me give you an example of a study that we just actually had come out last week, which is a collaboration with Christopher Fiorilla's group at KAIST in Korea, which is trying to figure out how do we enumerate the signals in the brain that can drive positive things like reward and pleasure, but when hijacked can lead to addiction and other negative states. And what we did was we used one of those targeting tricks basically to make different cell types. In this case, one particular cell type is what we focused on for this study, the dopamine neurons, which you might have heard of. They're often referred to in the media as some of the pleasure centers of the brain. And we equipped a mouse so that if the mouse went to one spot, it would get a pulse of light, and if the mouse went to another spot, it wouldn't. And so if basically the mouse goes to a spot and gets a pulse of light that activates these dopamine neurons, which we've made sensitive to light, and they will work for the light, that highlights a potential constraint on the circuit that allows us to figure out what properties of it might lead to an addictive state. And so here you can see a box where there's one spot where the mouse gets light and one where it doesn't. And here's that mouse, which is going to that spot, working for the light, if you will, over and over again. And so this allows us to pinpoint a place in the circuit and also the temporal requirements for which it is driven in order to result in what might be reminiscent of an addictive state. And the mouse will not go to the other site because it will not get a pulse of light at that site. So this is of interest to us because it potentially will allow us to screen through the brain, looking for neural targets that might be of importance in the way that they contribute to a brain disorder state, or that allow us to screen for sites in the brain that when we actuate them, we actually can figure out if we can remedy a brain disorder state. And so in a collaboration with Cliff Fonstad's group here in EE at MIT, we're actually building arrays of waveguides, little tiny optical fiber-like devices that can be in the many thousands or maybe even tens of thousands that allow us to tile the brain with light in a three-dimensional fashion. And one of the hopes is that we could actually do high-throughput screening through the brain, looking through all these points in the circuit for which points can contribute to a disease pathology state or which, when actuated, can remedy that disorder state. And if we find sites in the brain that, when we perturb them, allow us to fix a brain disorder, remember we have that robot that can go in and figure out what the molecules are that are associated with that circuit. And that, of course, could let us figure out what drug targets we should be looking for in a pharmaceutical sense. Now, there's another reason that we can use these prosthetics directly, perhaps, in the human brain. Invasive prosthetics are in widespread use. Perhaps 250,000 people have some kind of implanted electrical stimulator already. And if you think about it, we have to get the genes for these light-activated proteins into the brain cells. But as you've seen, we were able to do that. And the viral vectors that we use to deliver the genes are in widespread clinical exploration, at least at the clinical trial level, where they appear to be relatively safe. And so the question now is, what is this going to look like? Well, we and our collaborators here at MIT and at other places are doing preclinical tests to look at the safety and efficacy. And I'll tell you one last story about how we're doing that in a second. But before I get to that, I want to point out that Roz mentioned epilepsy in her talk. One of the great hopes is that we could actually turn off epileptic circuits right when they occur. And to go after this, we've actually gone back to the tree of life to look for molecules that pump electricity the opposite way of the one that we just showed you. So the one we showed you before converts light into neural activation. Over the last several years, we've found several classes, actually, of molecules that will actually allow us to shut down neurons in response to light. And we just received a grant to actually pursue this, where we are now looking for targets in the brain that, if we make them sensitive to light in the negative sense, where we can turn off those overactive cells, can we shut them down just during the duration of an epileptic seizure, let's say, so that we can do more efficacious and more side effect-free therapeutic strategies. Because for now, if epilepsy fails at a drug level, then often people have to seek neurosurgery to remove that region of the brain permanently. Another thing that we're doing along this solid clinical path is to try to figure out how to build fully wireless optical stimulators as well, things that don't require batteries, that can be remotely powered, that will make the realm of clinical prosthetics more real. And so one of the things we've done is to build a small device that contains all within it, the light sources, the power circuitry, an optical fiber, and weighs a few grams that can then be put into a living nervous system to allow us to fix a brain disorder in the fashion that you might see a pacemaker being used for the heart. Now, I want to end on one concrete last story, which is an area where we had a paper come out in the middle of last year, which is to try to explore how you might actually remedy one area of disorders which has been very prominent as a need that we would like to be able to address. And that's several forms of blindness that affect perhaps over 10 million people around the globe, in which the photoreceptors, the cells in the back of the eye that convert light into signals that the brain can understand, are lost or atrophied. Now, one of the nice things to know is that the rest of the eye, the other cells or neurons in the eye that aren't the photoreceptors, are often spared, sometimes for many, many years, sometimes for the rest of the lifespan. And so one idea, let's zoom in on this retina now so we can kind of see the circuitry a little bit. At the top are the photoreceptors. Those are the cells that capture light and turn them into signals that are then transmitted through a whole set of other cells into forms that are processed. You know, there's some motion detection, other things that go on. And then finally, the ganglion cells, the lowest level cells that you see there, will relay these signals into the brain. And in patients who have lost their photoreceptors, basically that initial light capture step is gone. Now, of course, the eye is still there. You know, light can still get in, right? And so one of the ideas that's been popular is to try to electrically stimulate the cells, but the electricity kind of spreads out. And so what we've been focusing on, along with collaborators such as Alan Horsager's group at the University of Southern California, is to make the cells in the eye photosensitive through the methods that we just described. This is also a topic of a startup company that we've co-founded. So how does this work? Well, let's show you some actual preclinical data that we found in our study last year. What we did was to take mutant mice that are naturally in mouse colonies throughout the world. You see them at pet stores even. And they're blind. They have a mutation that causes the photoreceptors to degenerate. And we put this mouse into a maze with 6 arms, so the mouse has to navigate the maze. And as you can see, a blind mouse, the mice are smart, so they eventually do solve the maze, but they're not using vision to do it. They have to kind of hunt around all these arms in order to find the way out of the maze. In this case, the maze has a little bit of water in the maze to motivate the mouse to swim. Mice like to swim, but they like to sit more. And under a lamp here is a small platform where the mice can crawl out of the water and escape the maze. So here's another mutant mice strain, again, not using vision, and eventually will solve the maze through brute force search, but isn't using its sensory apparatus to navigate. So this mouse was blind several weeks before, had 1 dose of these light-sensitive molecules encoded for by the gene therapy vectors, and is now able to navigate towards the platform. It doesn't prove that these mice have vision the way that you or I have vision, but it does show that these mice can make cognitive use of their visual information. And so we regard that at least as a step in the right direction. And if the science continues to be borne out through the safety and efficacy studies that we're continuing to do, then we're hopeful that this might lead to new kinds of prosthetics that allow us to do ultra-precise targeting of specific kinds of cell, even deep within intact tissue in the nervous system. And there's too many people to thank, but I'll put this slide up, and I guess if there's any questions, we'll end there. Thank you.
So, Matt, can you put that video up one more time? That last video, just up one more time. If you could just talk us through this one more time. It's extraordinary because the level, all right, this is treated rat.
That's right.
Immediately going to the light sensitive.
It took some training, actually. So much like cochlear implants require some usage, the mouse had to kind of relearn to use its eyes. It had to kind of practice navigating the maze. And over a period of about a week or two, it really learned how to navigate, again, using visual cues.
What did you learn about how that encoding process takes place? In other words, as the mouse gets used to its new light-sensitive cells on top of the photoreceptors, or whatever the ganglia are, as they get used to that, how do they understand that that's visual information, that relates to a state in the outside world?
It's a great question. So to be upfront, I'm not sure that these experiments totally prove the case all the way out to that they're making sense of the visual world in the way that we make sense of the visual world. One could imagine that they're just cleverly using shadows and brightnesses to help navigate towards the platform. A more sophisticated experiment, I think, which we are now trying to figure out how to do, with symbolic representations, let's say, scattered about, would allow us to pinpoint whether they're able to really detect form. Nevertheless, there was a training process where they had to kind of learn, through trial and error, kind of what made sense and to associate the path out with what they are visually encountering. And so we were still able to see that they had to take, there was some plasticity that was required over a period of many days in order to re-encode the visual world in this representation.
But once they get it, then they immediately know where to go.
That's right. So the video there is shown after the animals have learned to master their repaired eyes and are able to navigate very quickly. And they can actually navigate towards the platform just as fast as normal mice that have seen, as long as a normal mouse does.
Let's talk a little bit about your light-sensitive viral expression genetic therapy for activating certain kinds of cells and giving you a channel in to switch them on and switch them off. It is evident to me that most of us do not have clear plexiglass craniums, and that the light source within the brain to actually do what it is that you want to do would be a non-trivial matter, I suspect. Talk a little bit about the requirement that it be light. And of course, there are other radio frequencies or other RF energy that can penetrate the skull that could do similar kinds of activity. What are you looking at there?
Yeah, it's a great question. So there are many forms of energy that can be used to interface to the brain and body. And we and many others are actually looking at everything from ultrasound to magnetic fields, electricity, to allow us to interface towards what these cells do. Light, though, has a couple features that make it still very apropos to what we would like to do. First of all, it can be focused. You can stimulate with those 1,000-channel arrays that I showed at the end. We're really hopeful that eventually we could interface to the single-cell level, making brain coprocessors that can sort of speak the language of the nervous system at the single-cell level. With magnetic fields or radio frequency, there might be a little bit less ability to do detailed channel sharing because magnetic fields will spread out. If you put a barb magnet with some iron filings, you'll see the fields go in loops that are hard to focus. And so we're exploring these different possibilities because we think that they're going to be useful for different areas in the clinical space. So if there's an area of neurology or psychiatry where the goal really has to be addressed by very intimate, detailed interfacing at the single neuron level, then light might be the way to go. But some of our collaborators are already exploring how magnetic pulses, delivered even non-invasively, could be used to activate or perturb neural circuits. And in fact, we have a new research fellow who's going to be arriving this summer to try and figure out if we can close that gap between magnetic and optical stimulation and even unify some of these pictures, perhaps.
Finally, this is the part of the presentation where I want to get a window onto the Ed Boyden brain, not just brains generally. What's your theoretical supposition? And, stop me if I'm pushing you into a scary place here, but if we look at that small bit of brain tissue and try to imagine what the parts list is in a cubic millimeter or a cubic nanometer, whatever scale you want to talk about, what do you think is going on there? Is it a set of Lego-like parts that interact with a few sort of types and then that's sort of replicated around the brain and works in a sort of massive network? Or are we talking about very specialized function computing arrangements that move as we move through the regions of the brain?
Well, there's obviously a lot of specialization in different circuits in the brain. The degree to which we can learn about one region of the brain and then generalize to others, though, is still kind of one of the big open questions. I think we're going to find a lot of very detailed, specific computational motifs that are going to be very important to sort out. And I think the parts list is only the beginning. Those parts are going to change state, with learning, with the progression throughout life, with disease states. And so, we might find out that sort of like a nonlinear dynamical system where, the points in it orbit around. We've all seen those Lorentz attractors where, the chaotic weather patterns emerge from very simple equations. We might find that the parts list of the brain is a moving target, that within seconds they're reconfiguring, that within minutes they're changing in a plastic way, and that over the timescales of hours to days they rewire completely as gene expression and longer processes occur. So I wouldn't underestimate the brain.
No, and nor yours as well. Ed, thanks so much.
Thanks so much, Dave. Appreciate it.
For the past 20 years, our next presenter has been a professor of biomedical engineering and neurobiology at the University of Southern California. He's also director of the Center for Neural Engineering at USC, a position he's held for almost as long as he's been at USC as a professor. He's a pioneer in understanding the brain, both in his experimental methods and his theoretical basis. Please welcome Theodore Berger.
I hope this is the right one. Hello, and I want to thank the previous speakers for terrific talks. I'll do what I can to compete in a nice way. I actually had a different title, but Ed gave me this one, and I thought his title was better than mine, so I kept engineering memories. It's really good. So I do want to take off directly from what Ed was talking about, and that is the development of neuroprostheses and coming up with technical and theoretical ways of bidirectionally communicating with the brain to understand what or at least to be able to sense what the brain is telling us, and then having us develop the tools and the techniques for talking back to the brain. And I want to talk I can do this, I'm sorry. We've heard about the artificial retina and other types of prostheses, so I will, and if Hugh was up here talking, we'd hear about motor prostheses, so you're all oriented toward that. I'll skip that. And what we're working on is qualitatively different from, I would argue, is qualitatively different from sensory and motor prostheses, because we're working on parts of the brain that are central. That is, there are many synapses removed from the sensory parts of the brain, and there are many synapses removed from the motor parts of the brain. So if we discover that there's a part of the brain that's been damaged, my little cartoon here has brain area B, and if that's been damaged, then we need to have some way to model, to mimic how that brain area B received signals in neural terms, not in our cognitive terms, but in the neural terms of the brain. How does it receive signals from brain area A, transform them in the way that it normally does into different output signals, and then pass it on to brain area C? So we've got to be able to sense in the neural terms, and we have to be able to talk back to the brain in neural terms. And I'll show you that we think we can do this, and we are trying to prove to our satisfaction and to others that we can do this for a particular cognitive function, and that cognitive function is memory. So we're working on a part of the brain that's called the hippocampus, and what it normally does is to transform short-term memories into long-term memories. So if everything else works fine, and you're able to use sensory parts of the brain to essentially analyze what's going on in the external world, that can be passed through very complex circuits, but nonetheless identifiable circuits. And there's a pattern of activity that appears in areas that are afferent, that are the inputs to hippocampus, as a short-term memory. So, and that will stay a short-term memory and nothing more than a short-term memory unless it passes through the hippocampus. And the typical disorders that prevent this information from passing through the hippocampus are epilepsy, aging, stroke, and even blunt head trauma. And what we're proposing to do is to identify, both to characterize experimentally and then to model how parts of the hippocampus transform these inputs into outputs. And then we're installing and instantiating these algorithms into hardware, which is then first software and then hardware, either in terms, well, I won't tell you, you know what forms of hardware can be, and then we do that so that we can miniaturize this transformation, and so that the patient can essentially carry it around on him. In this case, the patient is a rat, but nonetheless can carry this transformative signal transformation along with it, and then we look at the output of our device and drive the output of the hippocampus into the appropriate output for a given input. So as was just pointed out in the last talk, this is not trivial. It has to be done in real time, and it has to be done from multiple pathways. So, and I'll show you how far we are in that. There have been a lot of problems which we had to overcome to mimic this short-term memory to long-term memory transformation. We are analyzing this entire circuit, but I'll be concentrating today on what's labeled CA3 here and what's labeled CA1. These are just two parts of the circuit. They happen to be at a certain, they're composed of many cell types, as you just heard, but the principal cell type, there's a principal cell type that receives information from upstream areas and then transforms that input and passes it on to downstream areas. So we're looking at that particular part of the circuit as just an example of how neural signals are transformed. So what kind of transformations are these, and and what are the problems associated with them? Neurons communicate with each other using all or none pulses, or what are called action potentials, but they're all or none signals, so the amplitude of that signal can't carry information. It's the timing. It's the time between spikes, what are called spikes, that carry information. And so it's what you can conceive of neurons, I think, very accurately as units that transform incoming temporal patterns into different outgoing temporal patterns. And because most things are coded in the brain by populations of cells and not by single cells, tonight when you have a beer or a glass of wine, you'll kill a couple 100,000 neurons, and yet you won't be a dramatically different person tomorrow. So single, well, sometimes. It's really distressing when it does happen. But things are represented by populations of cells. Single cells don't really contribute that much in and of themselves. So we have to think in terms of populations of cells transforming incoming spatiotemporal patterns into different outgoing spatiotemporal patterns. And there are a lot of mechanisms that are responsible for this nonlinear transformation. I won't talk about them, but they're clearly identifiable. We know what they are, and we can manipulate those. And so we know what the basis of this nonlinear transformation is. We'll skip this. The only thing I'll say, so the kind of task that we and a lot of other people use when we're investigating long-term memory formation by the hippocampus is something called a match to sample or a non-match to sample. We basically show the organism something and then take it away. So the organism has to solve the problem, to get a reward, the organism has to remember what this thing was that it saw, and then either choose it again or choose something different from that. And so we do this with rats. They face a wall and we show them one of two things in this case, and then the rat has to go do something else, poke its nose in a hole in the opposite side of the cage. I don't know who thinks these things up, but I do them because other people do them. And they seem to work really well. I mean, the rat has to remember this thing, and so it comes back to the same wall, and two things come out of the wall, and it has to press the opposite of the one that it pressed before. So the animal can show you that it does form a long-term memory, and long-term memories to a rat are anything in excess of 10 seconds or 15 seconds. That's what long-term means to a rat. And so what this slide shows is that as the time is extended, the blue line goes down. That's just the forgetting curve of this animal for remembering one out of two things. I don't want to spend that much time on these things. Anyway, there are specific modeling methods that we've developed and applied. And this is a real, obviously this is, and the important part of this is the modeling methodology keeps track of when things occur with a millisecond time basis. So the kernels of this equation are the representations of what happened to the output when we had this pair, what happened to the output when we had this triplet, what happened to the output when we had all the things in between. So it can be applied to spike-trained data like we're getting from the animal, and that's what we do. When the animal's learning this thing, we have a set of electrodes that are in this CA3, CA1 connection, so they're in line with, how do I go back? They're in line with the circuitry, which is really important. There. Sorry. There. Okay. Don't change. Okay. So this is just an example, a small example of a number of cells that we recorded from this one population that's upstream. So it's receiving multiple spike train inputs, and the region is then transforming that spatiotemporal pattern into a different one. And the methodology that we use has turned out to be very accurate for looking at a sample and being able to identify the critical spatiotemporal patterns and being able to predict what's going to happen next on the basis of the current spatiotemporal pattern. And so there's one of these is the real and one is the predicted. You can see that they're close. So we are able to find these patterns, and what's important is that these spatiotemporal patterns are different for different things. So in what's your upper left, that's what the pattern for a small population looks like when the lever or the thing is on the left, right? When the lever or the thing is on the right, you get a pattern that's shown below on the lower left. So different things result in different spatiotemporal patterns. That doesn't surprise you, but this is actually the first kind of data. In fact, it is the first data that shows that for memories, for long-term memories, there are different spatiotemporal patterns that can be clearly identified from rat to rat and from time to time. And so when we can look at these spatiotemporal patterns and predict what the animal is looking at or what the animal is doing, and we can also predict, for example, when the animal is going to make a mistake. If we show him something on the left and what the animal encodes is something on the right, then he's encoded the wrong thing, and when he comes back, he'll make a mistake. And we can show that that's the case. Okay? But these are the patterns that occur repeatedly when the animal is learning this task. So what do we do with it in terms of a prosthesis? Here. So we put animals in this task, having to learn whether something was on the left or the right. And we recorded these position-specific and task-specific and all kinds of other specific spatiotemporal patterns, and we saved them. Then we introduced, through a small mini-pump, a drug which blocks one of the critical channels. And this is the kind of specific you probably don't want to know about, but nonetheless, we can introduce a drug that silences most of the neurons, or at least slows them down a lot. And it also makes them, for the occasions when the cells are active, they now generate different patterns than they used to. So the memories, the sort of long-term memory that's being pumped out of the hippocampus to other parts of the brain is now much smaller, and it's wrong, right? So the animal shows us that it doesn't have a long-term memory anymore. It can't remember anything, in fact, past 10 seconds. It's too bad for the animal, but we now get to fix it. And the way we fix it is by when the sample is presented to the animal and he's supposed to generate one of these spatiotemporal patterns for long-term memory, he can't. So we do it. We already know which sample, what the spatiotemporal pattern is supposed to be, so we electrically stimulate through the same electrodes that we used to record from. We introduce the spatiotemporal pattern for left, or the spatiotemporal pattern for right. And what happens is then the animal shows us that it has a long-term memory. So we can reinstantiate this long-term memory by putting in these signals that tell the rest of the brain what it is that's supposed to be remembered. And on a group basis, it looks something like this. So this is the forgetting curve, and with this drug, the whole curve falls down, and in fact, the animal's at chance levels and choosing past 10 seconds. And then if we introduce the electrical stimulation, we push that curve back up. So, and we can, I can't show you here, but we can, in fact, give the animal the wrong memory, and it'll come back and make a mistake. So we can give them either the right one or the wrong one, and introduce this capability for re-encoding short-term memories into long-term memories. I won't go through in detail much about this, but if the animal's endogenously generating these spatiotemporal patterns that create long-term memories, then why don't we stimulate at the same time and enhance the signal, this long-term memory signal? And if we do that, we enhance the animal's capability for long-term memory. So the forgetting curve shifts from the blue up to the red. So in times and cases when the animal would otherwise be at chance levels, he's now firing very, very accurately, or behaving very accurately. There are lots of different controls for why this is due to the specific patterns, which we have, but I don't want to go through it, and I'm too long. So the bottom line is that we think that by paying attention to the specific spatiotemporal coding capability that's going on there, and we're assuming that everything in the brain is coded differently in terms of spatiotemporal patterns, that if we can identify, if we can mathematically model the nonlinear transform that makes short-term memories into long-term memories, or that makes anything into something else, that's a cognitive action or a cognitive operation, that we can reproduce that cognitive operation and reinstall, basically, that cognitive operation into an animal that otherwise has damage to the brain. And these are only some of the people that did this, and of course, money. So thank you.
I'll have some questions for you when we do the panel. Let me introduce Sebastien, and then we'll get you guys up here with Marvin Minsky at the end of this session. But thank you so much. That was terrific. His background is a PhD in theoretical physics from Harvard. He's the author of the highly acclaimed Connectome, How the Brain's Wiring Makes Us Who We Are. Our next presenter is Professor of Computational Neuroscience at MIT to talk about what we find in the computational world to produce insights in neuroscience. Please welcome Sebastien Seung.
Thank you, John. Thank you all for coming. So how many have heard of this new word, connectome? Could you raise your hand? All right. I want to congratulate you. You're on the cutting edge, you're a leading edge intellectual in the avant-garde. But for those of you who didn't raise your hands, I don't want you to feel bad because this is a new word. So you have an excuse as to why you haven't heard it yet. And not only that, you can go away from this gathering and go to cocktail parties and impress your friends by telling them about the next revolution in neuroscience. So John referred to the book that I wrote. I spent 3 years of my life off and on writing a book called Connectome. And the reason I did that, people often ask, why would you waste your time as a practicing scientist doing something like this? It's a lot of work. And the reason is I started on this path 6 years ago, and I encountered a lot of resistance from my colleagues. So the two kinds of objections that I got were really either about the how or about the why. So the first one was, you'll never be able to do it. It's too difficult to map out a connectome. And I forgot to explain what a connectome is. So a connectome is a new word. It's the wiring diagram of your brain, right? All the connections between all the neurons. It's been coined in analogy to genome. And so they said, it's too difficult to map this out, and so you'll never succeed. Now, that's okay. I can take that. But there's another more clever objection, which is about the why. They said, well, even if you succeed, all that data will be useless. Ooh, So, so I wrote this book to explain how we're going to do it and why we're trying to do this. And it's been getting some exposure. Some people are reading it, but I still have a lot of objections from my colleagues. A lot of people still tell me that this is not the right thing to be doing. And of course, a lot of them are saying it behind my back. So, because some scientists do have social graces. So the thing is that I've been thinking about this, and what's the proper response? And over the last year, I've been realizing that the world has changed. I don't need them to succeed in mapping connectomes. I actually need you. Not only that, I need the world. Because the technologies we have today allow us a new model of doing science, of open innovation, to get everyone involved. And if you think that I'm exaggerating by saying the entire world, remember that the brain is awesome in its complexity. How can we hope to succeed if my lab is the only one working on this, or even the community of neuroscientists? We need everything we've got. And so this talk is about calling on you, technically minded people, and also the people out there to come help us. I'm going to explain why we're trying to do this and how we're going to do it. And I'll give you an example, just to motivate you, what we could achieve. Last year, I met two wonderful women who run this foundation, the Phelan-McDermid Syndrome Foundation. It's a very rare genetic disorder. Recently, this disorder, it's not that many patients suffering from it, but this has been thrust into the spotlight in science because deletion of the SHANK3 gene, it's a chromosomal deletion, and in particular, deletion of a gene called SHANK3 is thought to cause this syndrome. And one of the symptoms, there's a lot of symptoms, but one of them is autism. And this finding of the genetic abnormality associated with autism has led to work by one of my collaborators at MIT. Actually, I want to collaborate with him because he's a great guy, but we haven't raised the funding yet. But I admire his work, and he's shown that if you damage this gene in mice, it produces symptoms that are reminiscent of autism. I mean, so this is the mouse model of autism, and I don't want to overplay it because can a mouse really have autism? Mice are different from humans, but it's intriguing that mice with this genetic abnormality have asocial behavior. So if you put them in a cage with a strange mouse, they'll stay on the other side. And they also exhibit repetitive behaviors. So this mouse will groom itself excessively until its skin gets irritated. And so what my colleague at MIT has found is that SHANK3 is highly expressed in the striatum, a part of the basal ganglia, which is implicated in many different kinds of brain disorders. And so the natural question arises, what's wrong with the striatum in these mice? What could be causing them to behave differently than normal mice? Could it be that this brain region is wired differently? And you hear that a lot in the media. Johnny's just that way because his brain is wired differently. Right? You've heard it all the time. Sepp Kamvar said, because people are wired to do it this way. But in reality, those assertions are faith. They're not based on evidence because we've never been able to see the wiring clearly. Just to give you some element of plausibility, I mean, you've all seen this kind of wiring mess. What's in your brain is far more complex, and it's certainly plausible that there could be miswirings of the brain. But in order to see those miswirings, we have to have imaging that's precise enough, that has sharp enough resolution to resolve those tiny wires. And only recently has that become possible. Here's how it's done. Imagine a piece of mouse brain. This is a piece of mouse brain. It's embedded in a very hard plastic. It's inserted into a really high-tech deli slicer. This cuts slices that are 1,000 times thinner than a hair. They float off onto the surface of the water inside that blue tub, and they're collected on this white conveyor belt. You can see little smudges of brain tissue that are being collected on the tape. And then we glue those strips of tape to a silicon wafer and place them in an electron microscope. And we're zooming in by a factor of 100,000 times to see what the brain really looks like at the nanoscale. These images come from the laboratory of Jeff Lichtman, my collaborator at Harvard. Now, 2 dimensions is not enough. That's just one image. We can take multiple images of many slices and stack them up to make a 3-dimensional image. And still, this may look complex to you if you've never seen it before. So to guide your eyes, we can just color in the cross-section of one branch of a neuron and do that over and over again, slice by slice by slice through this volume. And here we see revealed a reconstruction of a dendrite. You've heard about dendrites, and here it is with lots of spines. That's actually a very short segment of dendrite. And in the green, there is a short segment of an axon, and they touch at 2 points. Those are synapses. To verify their synapses, we zoom in and look inside the green neuron. You can see little circles. Those are called vesicles. They contain neurotransmitter. You've heard about dopamine or glutamate and so on. When the green neuron wants to send out a message, it spits out the contents of one of those vesicles, and the red neuron receives that. So what you've seen in these pictures is that if we have really clear images of the brain, then we can not only see the synapses, we can see the places where neurons connect. We can also trace the pathways of those wires through the brain, the branches of neurons. And so if we can do that, we can find a wiring diagram or a connectome. And maybe we could find out whether the brain is wired differently in autism. What's the catch? Sounds great. Well, this wiring diagram is the only one that is known in its entirety. It's got 300 neurons and 7,000 connections, and it comes from this humble worm. It was determined in the 1970s and 80s, and it took over a dozen years of effort. How could we hope to find connectomes of pieces of mouse brain or human brain if we can't even do this? Well, the technologies have been getting better. This is a new microscope. Carl Zeiss Incorporated is developing this. It will be delivered in 18 months to Harvard University, the first customer. It will generate 1 petabyte every 2 weeks. It has 61 beams that scan in parallel to collect data 61 times faster. That means we'll be able to image. We'll have the data in our hands, or at least not in our hands, in our hard disks. What is 1 petabyte? 1 petabyte is 1 cubic millimeter of brain, not a whole brain, but just 1 cubic millimeter. Remember, that 1 cubic millimeter is 1 pixel in an MRI brain scan. But that pixel contains a universe, 100,000 neurons, 1 billion connections. And let me tell you the truth. That fancy graphic I showed you with the coloring of the neurons, that was done by hand, by Daniel, a postdoc in my laboratory. And I calculated if Daniel were to color this petabyte in, it would take him over 100,000 years of work. That's what we're up against. What can we do? Well, we need AI. We need artificial intelligence. So far, we can speed up this process. By having humans interact with AI, we can go 10 to 100 times faster. And I hope that that pace of innovation will continue. Maybe some of you here will help make the AI better. But we also need people. We need people to interact with the AI to fix the mistakes of the machine. And not only that, to teach it to become better, because of course the AI that we have is created by machine learning. And we need more data. We need more data to feed the AI to make it smarter. So to do this, to try to explore the idea of inviting people to participate, we've created a website where people can come and participate in mapping of connectomes. So this website is called iWire. EYEwire.org. We're not studying autism yet, but we are studying the retina, the sheet of neural tissue at the back of the eye, which Ed talked about. And in fact, the volunteer spirit worked here. A bunch of MIT students taking my IAP class helped us make this website. And Elizabeth wrote the call to arms. Welcome to iWire, where you can help make discoveries about the neural structure of the retina. They wrote educational materials like this page about neurons and about the retina and so on. And two developers in my lab, Matt Balcom and Mark Richardson, have been working on this game to make it fun for people to help us map out connectomes. And you can see that 15 players are currently online. This game has been online. We were in private beta for 2 months. We've just gone into public beta. And here is the neuron that's currently being built. And you see these green cubes right here? These green cubes are of the same size as the cube that you saw before. And each of those is a location where one of those players is tracing out one of the wires of the retina. You can imagine them as little people climbing over this tree and extending its branches. And on the right-hand side, you can see the leaderboard. And if we click on one person, we can see their contribution to this endeavor. So you can see that the people at the top of the leaderboard have been really busy. They've been helping us map the connectome. And I'll show you the game, too, from the perspective of a new user. It's very simple. It's a 3-dimensional coloring book. A new user logs in and gets this instructional video, and then they get a little cube of the retina. You can see the cube over here on the left. And this is an image, one slice out of that cube, and we can move the yellow plane up and down and see where the AI has colored in part of a branch. And equally well, we can see where the AI should keep on going. And if we click right here, we can make the AI continue. And there we see that the AI has traced out the rest of that branch. Very simple. First empirical finding. Some people find this fun. People on the leaderboard are addicted. They're playing this game. There's something elementary about it. At some point in time, I think all of us enjoyed coloring. And some people say it reminds them of a lava lamp. Sort of organic forms, sinuous twisting. Something about that is meditative or relaxing. Now, I'm not saying this is a really slick shoot-'em-up game, which really is addictive, but this is just the start. This is kind of fun. It's kind of fun. There's a competitive spirit. And so in order to learn more about our users, we made a forum, a discussion forum. And as you can see, it became very active very quickly. And so I'd like to show you a post from Jose. Jose he was #1 on the leaderboard for weeks. And he's from Spain. And he tells us that he plays with his 7-year-old daughter. I had no idea. It's a family activity. And then he said, and look at this piece of a neuron. It looks like an ice skater. And I was shocked. I was like, wow, because the game, we got out of the game what we put into it. But now we start to see the creativity, things that surprised us, things that people came up with that we never expected. So continuing in that vein, another user said, well, I give you the finger. So we realized we should make a gallery portion of our site and allow people to post their pictures and caption them and vote them up or down and so on and so forth. But we haven't done it yet. We're too overloaded. We need help. We only have two developers doing this. So we'd love to do this. Now let me show you a more serious contribution. Of course, the bugs section of the forum is the one that has the most posts, sad to say. For several weeks, I was complaining to the developers that, the performance of this site is often bad. Sometimes the images get clogged, and it's really annoying. I can't play this game. The images don't load smoothly. And they said, well, it's your problem. Your internet connection is flaky. You have long latency packets. You have drop packets. We can't do anything about it. So one morning, I look online, and I see this post. And it's interesting. It starts out, I was having some long load time problems, i.e., I see the problem. And then iWire isn't making use of HTTP pipelining at all. Diagnosis of the problem. And then at the end, I'd also look into using a different proxy solution for the front end. I don't understand any of this, of course. Keep alive header, et cetera, et cetera. And then he concludes, just my 2 cents. He gave the fix. And I looked at my developers. They changed 3 lines, problem solved. They looked really embarrassed, but they were very happy. And so again, we saw the power of the community. And so that's how I became convinced, right? Maybe we don't need other PhD neuroscientists to succeed. What we need is to mobilize people like you and people out there in the world, anyone who's interested. I believe that we can create a community that breaks down the barriers between research, education, and entertainment. We could create a community that discovers what causes autism, learns about the brain, and has a good time doing it. And in the end, I would say that any activity in which we do all 3 of these things, we call that a quest. We do it together. It's an adventure. We learn something. We discover the unknown, and we enjoy each other. When I was a boy, I read about quests, mythic quests. Sir Galahad found the Holy Grail. And Ponce de Leon searched for the Fountain of Youth. Didn't find it, but he found an amazing new world. And I would argue that this is now the greatest adventure of all time, the ultimate brain quest. Please join us. Thank you.
Sit down, Sebastien. Theodore, come on up, please. And we want to welcome, who's been here all morning long, as someone who is very familiar to all of you, of course, the author of The Society of Mind and the Emotion Machine, long time, one of the spirits of the Media Lab itself, and also one of the founders of the entire field of artificial intelligence, Marvin Minsky. I have questions for all the panelists, but I also want to encourage you to get in on the act here. How are we doing, Joey? All right, good, good. You can expect to see some references from the conversation and some of the ideas that emerged in the presentations already in this session represented in some of the Google jockeying that's being done. First of all, Marvin, what do you make of the struggle to find useful metaphors for what goes on in the brain, either to describe the electrochemical processes that are taking place or some of the more high-level things that we understand that Roz talked about as personality and emotion?
It's a very complicated problem, and I certainly would love to know much more about the connectome. But we also need higher level theories to organize this, and I'm trying to think of a good example. There are billions of neurons in the brain and trillions of synapses, and one question is, how much of that do we need to understand? And every little bit of what comes out of this project is going to tell us not so much how an individual brain is connected, but maybe in the long run, the first few years of this is going to start informing us of things that we need not know about the same structures that you're seeing there. For example, imagine that a caveman were to look at a computer and say, well, in order to understand this thing, we're going to have to know what every atom is doing. And if you looked inside a computer and started at the bottom, then you would see these little chips, and in the chips you'd see these little bars of silicon, or used to be germanium, but they rusted. And in the silicon, you'd find a few parts per million of arsenic and phosphorus, or whatever they use to dope it. And so some people would say, the first thing we've got to understand is how a transistor works. And how come it doesn't work if it doesn't have just these very sparse impurities there, the arsenic and phosphorus or whatever it is. But if you take a higher level view of the computer, you'll notice that the computer never has a transistor except at the input and output. And they're always in pairs, and they form a flip-flop. Just as if you look in the cortex, yes, there are millions of synapses in every cortical column and maybe a couple of thousand cells. But maybe it's like a flip-flop. And what's the point of the flip-flop? It's to insulate how the brain works from how the neuron works, or from how the insulate how the computer works from how the transistors work. So what Sebastien has to do is, and gathering this data is the absolutely first and critical part, get the information about what's there, then, and only then, perhaps, can you start to look at all that data and say, what seems to be the important parts of it? Right now, it's just a guess. And I take the same view at the higher levels. If you look at the book I wrote in 1985, it talks about K-lines and paranomes and various things like that. And after a couple of decades of thinking about how thinking might work, I came up with about a dozen ideas for little structures that we probably need somewhere in the brain for doing higher level thinking. No neuroscientist has found any of those structures, and they don't know where to start. If I got even a coarse view of this connectome, I could guess where some of those are, and then we could have groups cooperating, the high-level cognitive theorists and the micro-level massive data collection would interact, and maybe a few years from now then we'll know exactly what to look for or more, and it'll only take 50,000 or 100,000 people instead of 20 million to process this data.
Theodore, how do the temporal spatial patterns that you're taking a look at in the experiments that you're doing constitute a language for understanding the states of the brain at this sort of mid-level that you talked about in your talk?
Yeah, very good. I agree completely with what you just said, that we need to have multiple levels of looking at brain structure and function. What we're trying to do is to develop a mid-level language of describing the status of circuits and describing them in terms of the nonlinear transformation that's produced by a collection, by a synaptically connected collection of neurons. And so whatever signals are generated when the rat or a human learns are the input, and how they become transformed into a different output reflects what kind of neurons are there and how they're connected and what the general state of the system is. So it does provide you with a description, with a quantitative description of how a population of neurons is interacting to produce this transformation.
Let's explore that relationship, because a lot of brain research that seems to be very exciting right now is fMRI, data, which if you look closely at it, even past the pretty pictures, and Joy, I'm sure you can come up with some pretty fMRI pictures for us here, and by the way, great stuff. It's really measuring hemodynamics in the brain. It's measuring blood flow. And the assumption is that there's some sort of useful relationship between blood flow and the actual neuronal activity, right. But it's probably not linear. And if it's not linear, it raises all kinds of questions about what it is that we're looking at. On your spatial temporal model, that you're looking at, do you believe there's a linearity in what's going on actually in the brain and the patterns that you're observing as you record off of these neurons, or is it not linear? Is it more like the hemodialysis or hemodynamics kind of problem with the fMRI?
I think it's definitely nonlinear. I don't think there's very much about the brain except the summation of ions in the extracellular space. I mean, that's a linear process. But everything else about neurons is clearly they're nonlinear signals, signal generators. They're very good. That's how I felt after flying from California yesterday. But they and I mean, again, you can measure it. I mean, it's so the current voltage relations that identify certain types of channels, they're almost all nonlinear channels. And the process of how one of those nonlinear channels interacts with another one is, of course, a different level of nonlinearity. And so there's no question about the fact that it's a nonlinear process from beginning to end, and complicated. I mean, so it isn't just a first order nonlinear, but second, third, and higher order nonlinearities. So
but the hope is that an emergent relationship that is predictable and that is robust enough to allow for very, very different kinds of neuronal functions will help us to make translations in different places in the brain. Sebastien, let's respond directly to the question that you raised first in your talk and which was amplified by Marvin here. If you had, stipulating for the moment that the difference between a brain that is dead and a processor chip that is off are profoundly different states, is wiring even a useful metaphor for thinking about the brain, in the sense that if you had a quad-core chip wiring diagram in front of you, not the caveman and the computer, would that wiring give you a sense of the energy flows and information transfers that take place on that chip, even if you knew the wiring from the diagram itself? Sebastien?
Well, I think that it would be important for an understanding of the computer, but I think that the computer may not be an extremely good metaphor for the brain. And the reason is that general purpose computers have a lot of the capability in the software. Right? A computer can be programmed to do anything. If you look at our brains, we have different parts which are specialized for different functions. So each part of our brain is a special purpose computer, which is why its wiring may be adapted to that particular task that it carries out. And the final thing I would say is that the wiring of the brain is not like the wiring of a computer because it changes. Right? So I call this the 4 R's of connectome change. Reweighting, reconnection, rewiring, and regeneration. So reweighting is a change in the strength of an existing connection. Reconnection is the creation of new synapses or the elimination of old ones. And rewiring would be the extension of new branches of neurons or retraction of branches. And regeneration is the most drastic change of all, which is creation of new neurons or destruction of old ones. So we can't use the hardware metaphor for the brain because the hardware itself changes.
So how does the connectome track those changing states?
At this point in time, we're looking for a snapshot. I'd rather have a really precise snapshot at this point than a blurry movie.
Marvin.
Speaking of that, there are all sorts of wonderful problems that come up with this. And jumping back to 1950, there was some discussion of if you turned a brain off, could you turn it on again? And this is rather mysterious, and John von Neumann was interested in this, and we had a discussion of if you had, this is almost before there were any computers, but if you had a computer, how would you start it up? And there was a contest to make a theory of how to, called the firing squad problem. How can you transmit a signal to get a lot of different things to work at once? And in fact, it takes a while to wake up from sleep. But the evolution of the brain had to
But this is not just a thought experiment. There are people who fall to the bottom of frozen lakes and their body temperature is so low that their brains basically shut off. There's no more activity left, and they're clinically dead. You pull them out, and sometimes they recover. And that's probably because the structure of the brain has remained intact, even though the signals have stopped. And doctors, amazingly, some doctors use this for surgical procedures, certain kinds of brain surgery. There's a procedure called PHCA, profound hypothermia and circulatory arrest, where the heart is stopped, the brain is cooled down, and people get woken up afterwards.
Let's talk a little bit about how the connectome as a snapshot could be a useful tool for describing some of the effective qualities that Roz was talking about if we can trace an emotion not simply to a sympathetic nervous state on the body, but something that's taking place inside the brain itself. How would the connectome help with that?
Well, right now, a lot of the study of the emotion in the brain has focused on where things happen. Where is emotion in the brain? Which to some extent we can answer, although maybe to some extent that's not the right kind of question. But the focus on this kind of really detailed resolution is not really about where. It's about how. How does it actually happen in the brain? What's the mechanisms by which computations or any kind of perceptions, thoughts, or feelings are taking place? And so that's what we're after.
Go ahead, Marvin.
On that, the distinction between emotion and intellect is an interesting one. And there's a general belief, I think, in the humanities that thinking and intellect is just sort of a matter of logic, and it's interesting, but it's not very complicated. You have propositions and you
nice.
rub them together. And emotions are terribly profound, and maybe it's just the opposite. That is, an emotional state is where some particular subset of the brain is running things, and it's running them in much the same way it would work in an alligator or not a sea anemone, but somewhere in between. And we have 1,000 words for different emotional states. And I suspect what we need is 1,000 words for intellectual states. When are you searching for a new hypothesis? When are you trying to find the difference between two things? And so forth. In my book, The Emotion Machine, there's a list of 30 or 40 different activities that we do have words for. And so this question of the humanities have grown up to worship emotions as being almost inexplicable, and thinking of thinking as just logic. And maybe it's just the opposite.
Yeah, by the way, if you haven't read Marvin's book, this is a really great, inspirational read. I enjoyed it very much.
And Society of Mind I also recommend as well, which really thought about a lot of these high-level issues very, very early on in the game. Theodore, let's talk for...
Who's doing that so quickly?
That's Joey. By the way, let's have a round of applause for the Google Docs up there. Our cognition, our sense of consciousness, Theodore, is a great metaphor for what we think is happening in our brains. We have an idea of what's going on in our own minds based on our personality and how we understand consciousness itself. How deceptive is that? In other words, does the brain distinguish between intellectual and emotional states, or is that just something we do as a part of this software called consciousness, and that inside the brain, these states have no distinctions that are terribly relevant at all?
Well, I think the evidence that's available today says that there clearly are parts of the brain that are active when there's an emotional response to whatever is going on. So there seems to be circuitry and neurons that are pretty much dedicated to adding that emotional content to whatever other analysis is being performed by other parts of the brain.
Although there's plasticity in the brain that people talk about in the sense that other pieces of brain tissue can take over functions that were once localized in a particular area, that suggests that we may be trying to recreate a sense of consciousness when there is a brain injury that results. But in fact, the brain is happy to reorganize itself in a variety of different ways.
I think that's a really important point, and it's relevant to the connectome issue, because that's even though we can identify connections that disappear and that reappear. There are functional states, there are functional properties, computational properties of different parts of the brain that are constant. So even though that at a micro level or a micro level, there are synapses that we can watch them grow and we can watch them be pulled back in. There's a global function which is clearly constant. And what governs that? We don't have any idea yet about what governs that constancy and how those rules are used or read out. And it's a really important issue which we don't have much insight into.
Final thoughts.
Popping
Go
back
ahead,
to
Marvin.
this emotion versus intellect thing. We see people in different emotional states and we identify different brain activities. But has anybody done an experiment where you confront somebody with Zeno's paradox and see what's happening and contrast that with something where you make a statement that's overgeneral and you need to be more... In other words, I don't see people doing experiments on particular intellectual puzzles, and that's sort of important because you can't get through the world without thinking, and nobody studies that.
And it's also, you know, interesting that in computer terms, division by zero is a fairly catastrophic event, yet in day-to-day life, it's a relatively trivial event. I don't think division by zero has stopped anyone from doing anything, probably in this room, but it can be catastrophic in machine terms, again, describing the important difference. We asked everybody this in the first panel.
In fact, when you get to a paradox, people laugh rather than explode.
which is great and very convenient because exploding people would not work. And imagine how many exploding people there would be at MIT if that was the way we responded to intellectual stress.
But maybe there's a joke so funny that they'll...
Yes. Well, for the Comedy Channel, perhaps, not for us. Then before we go to lunch, I asked everyone on the earlier panel, what inspires you most about nature, and how is that directed or realized in your own work? Sebastien?
Oh, what inspires me the most about nature? Nature's infinite variety, but also the networks of the brain and the amazing convergence of networks in our society. In some sense, the only chance we have of understanding the brain is to make ourselves smarter as people and as a society. And technology is making that possible through networks.
In design terms, what inspires you most, Theodore, about nature in your work?
The hierarchical organization of nature and of different systems. And the stability of the system despite this hierarchical organization. I mean, and I'm sure many people here have tried to develop a computer model of a system composed of complex, nonlinear, dynamical, smaller systems, and getting a system like that to be stable is not easy, and yet nature does it all the time, and does it apparently extremely well. I mean, there's a lot of perturbations that go on in daily life, and yet birds are not falling from the sky, and we in fact don't blow up. And it's the hierarchical organization that I really find striking, and its stability. That's amazing.
Marvin, take us out.
Yeah, making something with 1 trillion parts that lasts 100 years is pretty remarkable.
Love it. There you go. Can't do better than that, ladies and gentlemen. Have a great lunch. We'll see you back here for the last session at 2 o'clock. Listen for the signs that we're getting started in here because we're on a tight schedule in the afternoon. Thanks so much.